What's up, guys? It's Taysa and Avery back with our second presentation. And this week we are doing animal cloning, talking about the biotechnologies. So let's get into it. Animal cloning refers to creating a genetically identical copy of an animal. The most common method is called somatic cell nuclear transfer, which is also short for SCNT. Here's a breakdown of how that works.
How does it work? Cell selection. Scientists take a mature cell, like a skin cell from the animal they want to clone. Egg cell preparation. An egg cell from a different female animal. The original nucleus containing the egg's DNA is removed.
Nuclear transfer. The nucleus is carefully inserted into the empty egg cell. Fusion and development. The combined cell is stimulated to start dividing like a fertilized egg. This embryo is then implanted into a surrogate mother's uterus for gestation and birth.
Implantation. The cloned embryo is implanted into a surrogate mother's uterus for gestation and birth. Does it work? This process is complex and not very efficient. Dolly the sheep, the most famous clone, was just one success story out of hundreds of attempts.
Animal cloning has several potential applications. Agriculture. Cloning high-producing livestock for improved milk or meat yield. Research. Studying diseases and creating animals with specific genetic modifications for medical research. Conservation. Helping to preserve endangered species by reproducing rare animals. Companionship. Cloning a beloved pet for sentimental reasons.
Current use and success rates. Animal cloning is already being used, but on a limited scale. Here's a breakdown of current applications.
Research is the primary use for animal cloning currently. Scientists use clones to study diseases, particularly those with a genetic component. For example, cloned animals with specific gene mutations can help researchers understand how those mutations contribute to the disease.
Endangered species. While still in its early stages, cloning has shown some promise in aiding the conservation of endangered animals. Researchers are looking at using cloning to reintroduce lost genetic diversity into populations or creating populations where natural breeding is difficult.
Companion animal cloning is a more controversial use. Some companies offer cloning services for pet owners who wish to create a genetic copy of a deceased animal. However, this is a very expensive procedure with long ethical concerns, and its long-term health effects on its cloned pets are still being studied.
Animal cloning remains a complex and inefficient process. The success rate for producing a healthy clone is still quite low, typically around 1 to 4%. This means many attempts are needed to get just one viable clone.
Overall, animal cloning technology is still under development. While it holds promise for research and conservation, its widespread use is limited by the high cost, low success rate, and ongoing ethical discussions. Next, we want to talk to you guys about the pros and the cons of animal cloning.
The first pro is preservation of desirable traits. In agriculture, cloning can be used to reproduce animals with exceptional qualities like high milk yield or disease resistance. This can be helpful for improving food production efficiency.
The next pro of animal cloning is medical research. This is creating genetically identical animals with specific diseases, which allows scientists to study those diseases in a controlled way. This can accelerate advancements in medicine.
Another pro is conservation efforts. For critically endangered species with very few remaining individuals, cloning could offer a way to rebuild populations and preserve genetic diversity.
The last pro that we have is companion animals. Pet cloning offers emotional comfort to some who grieve a loss.
Now let's talk about cons. Animal welfare. The cloning process can be stressful and have health consequences for clones. Ethical concerns. There's debate about the ethics of creating clones solely for human benefit, especially for pets. Some argue it's unnatural. Low success rate. Animal cloning is a complex and inefficient process with a high chance of failure at each stage. Many cloned embryos don't survive to birth, and those that do may have health problems.
High cost. Cloning remains a very expensive procedure, limiting its accessibility and practicality for many applications. Uncertain long-term effects. The long-term health consequences of being a clone are not fully understood. Clones may have a higher risk of developing health problems later in life. Food safety. While regulatory agencies say meat and milk from cloned animals are safe to consume, some consumers are concerned about the potential unknown health risks.
Overall, animal cloning is a powerful technology with both potential benefits and drawbacks. As research continues, the success rate may improve, but ethical considerations and animal welfare concerns will likely remain a major point of discussion.
Animal cloning technology has the potential to be used for several other purposes beyond its current applications. Here are some possibilities.
Now we are going to discuss additional purposes. Xenotransplantation. This involves transplanting organs from animals into humans to address the shortage of donor organs. Cloning animals with human-compatible organs could be a solution, although significant biological hurdles remain.
Production of therapeutic proteins. Scientists could potentially engineer clones to produce valuable proteins used in their medications within their milk or blood. This could provide a more reliable and efficient way to manufacture certain drugs.
Models for human diseases. By precisely manipulating genes, researchers could create animal clones that develop specific human diseases. These models could be crucial for developing new treatments.
Reintroduction of extinct species. While technically challenging, cloning could potentially be used in efforts to bring back extinct animals, especially if combined with preserved genetic material.
The concept of a slippery slope is definitely a concern with animal cloning technology. Here's why. Progression toward human cloning, opponents worry that advancements in animal cloning could pave the way for human cloning, which raises serious ethical safety issues. Even if the focus is on therapeutic cloning, the technology could be misused for reproductive cloning. Commodification of animals. Cloning animals for commercial gain, such as high-producing livestock or creating designer pets, could lead to a disregard of animal welfare and turn animals into mere commodities. Unintended consequences. The long-term effect of cloning on animal health and ecosystems are not fully understood. Introducing cloning with specific traits could disrupt natural balances within animal populations. Unequal access and social justice. If cloning becomes more affordable, it could become a technology only accessible to the wealthy.
Here are some examples of how the slippery slope might play out. Cloning starts with the replicating high-yield livestock, then progresses to manipulating genes for desired traits, potentially leading to unforeseen consequences. Cloning for pet companionship becomes normalized, leading to pressure to create human clones for emotional attachment.
Steps to mitigate these risks. Clear regulations. Establishing strong international regulations on animal cloning can help prevent unethical practices and ensure animal welfare. Open discussions. Public discussions about the ethical implications of cloning are crucial to guide its development and use responsibly. Focus on welfare. Research efforts should prioritize animal welfare throughout the cloning process, minimizing any potential harm to animals.
The slippery slope concept is a major point of discussion in both scientific and political circles regarding animal cloning. The conversation generally revolves around two key points.
The risks of human cloning. Scientific concern. Scientists acknowledge the technical advancements in animal cloning could contribute to research on human cloning. However, the vast majority emphasize the significant biological hurdles that remain for human cloning. These include issues like placental development and immune rejection, making it a much more complex challenge than animal cloning. Policy debates. Despite the scientific hurdles, the potential for human cloning drives policy discussions. Many countries have already banned human cloning, while others are working on legislation to address these concerns.
Ethical considerations and potential misuse. Animal welfare. Scientists and ethicists discuss the ethical implications of animal cloning, particularly the potential impact on animal welfare. There's an ongoing debate about minimizing harm to animals used in the cloning process, including surrogate mothers and used for cell cloning. Focus on benefits versus risks. Discussions often center on balancing the potential benefits of cloning for research, conservation, or medicine with the risks of unintended consequences or misuse. Determining which applications are acceptable and which should be restricted is a complex issue.
Direction of the discussion. The overall direction leans towards responsible development. Here's a breakdown. Scientific research. There's a general consensus on the importance of clear guidelines for animal cloning research to ensure responsible use and prioritize animal welfare. Focus on specific applications. Discussions advocate for a focus on specific applications with clear benefits like medical research or conservation efforts, with strong oversight and regulations. International collaboration. There's a growing call for international collaboration to establish clear ethical guidelines and regulations for animal cloning to prevent misuse.
Legislation surrounding animal cloning varies around the world, with some countries having stricter regulations than others. Here's a breakdown of the current situation and arguments for and against stricter legislation.
Arguments for stricter legislation. Protecting animal welfare. Stricter legislation could ensure stronger safeguards to minimize harm to animals involved in the cloning process, including the surrogate mothers and cells used for cloning. Preventing unethical uses. Clear regulations could help prevent the technology from being misused for non-essential purposes, such as excessive commercialization or pet cloning. Addressing public concerns. Legislation can help address public anxieties about the safety of food from cloned animals and the ethical implications of this technology. Long-term oversight. As the technology develops, stricter legislation can provide a framework for ongoing oversight and risk assessment.
Arguments against stricter legislation. Hindering scientific progress. Overly restrictive regulations could hinder scientific research that could lead to advancements in medicine, conservation, or agriculture. Focus on enforcement. Some argue that existing animal welfare regulations, if properly enforced, can adequately address concerns. Innovation versus precaution. Strict legislation might stifle innovation in a rapidly developing field, potentially delaying potential benefits.
My take on legislation. While there are arguments against stricter legislation, I believe a cautious approach is warranted given the potential risks. Here's why. Unforeseen consequences. The long-term effects of cloning on animal health and ecosystems are not fully understood. Stricter legislation allows for a more cautious approach as we learn more. Ethical considerations. Animal cloning raises significant ethical concerns. Clear regulations can help ensure this technology is used responsibly and prioritize animal welfare. Public trust. Public anxieties surrounding animal cloning and the safety of cloned food products are real. Legislation can help rebuild public trust throughout transparency and oversight.
The debate on animal cloning legislation is likely to continue. Finding the right balance between encouraging responsible scientific development, protecting animal welfare, and addressing ethical concerns will be crucial.
Thank you guys so much for tuning in for our second presentation on animal cloning.
We really hope that you guys enjoyed this presentation, and please let us know if you have any questions, and if you do, drop them down in the comment section below.
And as always, don't forget to like and subscribe.